Good afternoon to all the students. So, today we are going to deal with the concepts nucleic acid and enzymes from the chapter Biomolecules. We are dealing with nucleic acids because the next sessions we are going to conduct are on Mendelian genetics, Mendelian disorders, for which you have to have a complete idea on the nucleic acids. Nucleic acids is a very broad chapter or a very broad concept in your both NCERT 11 and NCERT 12. If you are clear with the complete structural components of nucleic acid in NCERT 11 biomolecules, it will be easy for you when you are dealing with molecular basis of inheritance chapter in your NCERT 12 biology. So, nucleic acid, if we have to say, in simple terms, a nucleic acid is addressed as polynucleotide chain. It is addressed as polynucleotide chain. This polynucleotide chain can be ribose nucleic acid, we will call it as RNA, or it can be deoxyribonucleic acid, which we will call it as DNA. Both of them are polynucleotide chains only. If we have to take a single nucleotide, there are three components of a single nucleotide. One is sugar. What are those three components, ma? One is sugar, second is phosphate group, second you can take nitrogen base, and third is phosphate group. A single nucleotide consists of three components. One is sugar, nitrogen base and phosphate group. First we will see here you can see, right, a single nucleotide, it has how many? A single nucleotide, it has sugar, phosphate group and nitrogen base. What type of sugars you can see? You can see pentose sugar. That pentose sugar can be either ribose or deoxyribose. It can be ribose sugar or deoxyribose sugar. And nitrogen bases are of two types. We have both purines and pyrimidines. Purines are heterocyclic rings. It has two rings. Purines include adenine and guanine. Pyrimidines include cytosine, thymine and uracil. Cytosine and uracil will be there in RNA. Cytosine and thymine will be there in DNA. Phosphate group is added via phosphoric acid, that is H3PO4. Clear, right? And this phosphate group will give negative charge either to DNA or RNA. So, here we are clear, right? Nucleic acid is nothing but a polynucleotide chain. If you have to observe what are the components of a single nucleotide consists of a sugar, nitrogen base and phosphate group. Sugar is pentose sugar, it has 5 carbons. It can be ribose sugar or deoxyribose sugar. Nitrogen base can be either purines or pyrimidines. Purines, adenine and guanine. Pyrimidines can be cytosine, thymine and uracil. Phosphate group is in the form of phosphoric acid. This is about nucleic acids. Introduction of nucleic acids. Here we have taken one example by taking adenine nitrogen base, which is heterocyclic, this is purine, and deoxyribose sugar. Okay, we are taking deoxyribose sugar and adenine nitrogen base, right. So, here if you see nucleic acids, let us once go through the intro also. Together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. Nucleic acids are true biomacromolecules which are obtained under retentate when you do the chemical analysis. Along with polysaccharides and polypeptides, nucleic acids are also obtained in the retentate. Nucleotides are the building blocks of nucleic acids, and just now we have seen a single nucleotide is composed of what. And then three chemically distinct components here you can see. One is heterocyclic compound, the second is a monosaccharide, and third is phosphate group. Just now we have discussed. The heterocyclic compounds in nucleic acids are nitrogen bases. They can be adenine, guanine, which are purines, and uracil, cytosine and thymine, which are pyrimidines. Got it, ma? Now here you observe what are the bonds involved in the formation of a nucleoside and a nucleotide. First of all, what is the nucleoside? If sugar is adjoined only with the nitrogen base but not phosphate group, sugar and nitrogen base put together is known as nucleoside. To the nucleoside, if phosphate group is binded, then you will call it as nucleotide. To understand how the bonds are established between a sugar and a nitrogen base, sugar and phosphate group, first you have to know the complete 5 carbon pentose sugar structure, how many carbons are there and how are they arranged. These are the diagrams given in your NCERT structural diagrams. Now let me focus only on the pentose sugar. Here the diagram which is given is ribose. The structural formula includes C5H10 and O5. Here can you see, ma, the pentose sugar structure C1, C2, C3, C4 and C5. You have 5 carbons, right? Here in the ribose sugar at second carbon you have oxyhydroxyl group, okay? At second carbon you have hydroxyl group. The only difference between ribose sugar and deoxyribose sugar is, in deoxyribose sugar there is no oxygen group, okay? There is no oxygen group in deoxyribose sugar, only H is present. But here you can see the hydroxyl group at second carbon in the ribose sugar. That is the only structural difference. I hope now you got the clear structure of the pentose sugar. Now, how the bonds are established in a nucleoside. First, C1 to first carbon of the ribose sugar. If it is purine, N9 of purine is binded, and if it is pyrimidine, what are included under purines? Adenine and guanine. If it is pyrimidine, C1N1 bonds are present. C1N1, these are pyrimidines, which includes cytosine, thymine and uracil, okay? C1N9 glycosidic bonds, C1N1 glycosidic bonds are in between pyrimidines and sugar. So the bond established is C1N1 N9 glycosidic bond in between sugar and purine. It is C1N1 glycosidic bond in between sugar and pyrimidines. Clear, right? This is about the nucleoside formation. How a nucleotide is formed? If a phosphate group is attached to the 5th carbon of the nucleoside, then you will see the structure of a nucleotide. So here if you observe, to the 5th carbon, the phosphate group is established via phosphoric acid, that is H3PO4. The bond is formed. Which bond is formed here is phosphoester bond. Phosphoester bond. So here if you observe clearly, in a single nucleotide, at first carbon of the sugar, the nitrogen base is binded via glycosidic bond. At 5th carbon of the sugar, phosphate group is binded via phosphoester bond. So for a single nucleotide to form, 2 bonds are included. One is glycosidic bond and one is phosphoester bond. This bond is in between C1 and N9 of purine, C1N1 of pyrimidine, and the phosphate group is binded to C5 carbon of pentose sugar via phosphoester bond. Here if you observe the example which is given via taking adenine, nitrogen base, deoxyribose sugar, means at second carbon there is only H group and here phosphate group. Here can you observe clearly C1 and N9 glycosidic bond, C2, C3, C4 and this phosphate group is connected via C5. C5 carbon of the sugar is connected to the phosphate group. This is via N, this is via phosphoester bond. Clear with a single nucleotide? If you are having any doubts, you can drop a message in the chat box, ma. Now, we have observed the clear structure of a pentose sugar, right? Pentose sugar we have seen clearly. Now, let us look into the all types of nitrogen bases, both purines and pyrimidines. Very important, not just for the chapter biomolecules, but also for molecular basis of inheritance of your NCERT 12. Yeah, purines and pyrimidines. Can you see the heterocyclic rings of purines and homocyclic rings, single rings of pyrimidines? Their IUPAC names are different and you have to remember that, which I have tabulated here. Adenine, you can name it as the common name of the nitrogen base adenine, and this is the IUPAC name of it, which is 6-aminopurine. Okay. How is it 6-aminopurine? I will tell you. See, this is the first group, 3, 4, 5, 6. At 6th carbon amino group is there. So, the IUPAC name of adenine is what? 6-aminopurine. Next, guanine. Guanine you can say 2-amino-6-oxypurine. 2-amino-6-oxypurine. Here can you see 2, 3, 4, 5, 6. So, it is 2-amino-6-oxypurine. What is it? Guanine is 2-amino-6-oxypurine. Next comes to pyrimidines. Cytosine is also known as 4-amino-2-oxypyrimidine. Cytosine 4-amino-2-oxypyrimidine, right? 4-amino-2-oxypyrimidine. See, this is 4-amino-2-oxypyrimidine. And then thymine, it is 5-methyl-2,4-dioxypyrimidine. 5-methyl-2,4-dioxypyrimidine. 5-methyl-2,4-dioxypyrimidine, which is thymine. Okay. And then uracil, which is 2,4-dioxypyrimidine. Uracil is 2,4-dioxypyrimidine. Clear, right? Adenine is 6-aminopurine. Guanine is 2-amino-6-oxypurine. Cytosine, if you have to see, 4-amino-2-oxypyrimidine. Thymine 5-methyl-2,4-dioxypyrimidine. Uracil is 2,4-dioxypyrimidine. Very important slide, ma. The common names and the IUPAC names of the 5 nitrogen bases are very important. Okay. You can take a screenshot of this. Yes, you can take a screenshot of this and revise it very often. Right. And then now we have understood what are the bonds involved in a single nucleotide and how a single nucleotide is formed. Now we will see the chain of nucleotides. Okay, the chain of nucleotides. Here can you see there is one nucleotide at 5th carbon phosphate group, 1st carbon pyrimidine is attached, and this is a nucleotide. 3rd carbon has OH group, and to the succeeding nucleotide 5th carbon has phosphate group. Phosphate group of the succeeding nucleotide is binded to the preceding nucleotide via phosphodiester bond, because one ester bond is already there between phosphate group and carbon, right? And one more phosphoester bond is required to get binded with the third carbon and phosphate group. This is how two nucleotides are connected. So, two nucleotides are connected via phosphodiester bond. Phosphodiester bond. This phosphodiester bond is in between third carbon of one nucleotide to fifth carbon of the succeeding nucleotide. Now, for a formation of single stranded polynucleotide chain, how many bonds are required? That might be the question. Okay. To form a single nucleotide, what are the bonds included? We know. To form a single chain or single stranded DNA or RNA, how many bonds are included and what are they? If that is the question, your answer should be very clear because it is very simple. For the formation of single stranded DNA or RNA, 4 bonds are required. First is N-glycosidic bond. Second is phosphoester bond. And third bond is phosphodiester bond. These are 3 bonds, sorry. Phosphodiester bond. These 2 bonds are enough for the formation of a single nucleotide. Got it? For the formation of single nucleotide, these 2 bonds are enough. Right? So, this is in between first carbon to N9 of purine and N1 of pyrimidine. Clear, right? Phosphoester bond, it is in between fifth carbon to phosphate group. Okay, remember these 2. Phosphodiester bond is in between third carbon to fifth carbon of one nucleotide, third carbon of one nucleotide to fifth carbon of the succeeding nucleotide. Clear, right? Now, for the formation of a double stranded DNA or RNA, for that you have to know the salient features of double helical model which are proposed by Watson and Crick. First, who observed the double helical model? This is there in molecular biology. Let us go to some components of mol bio with which we can learn nucleic acid more clearly. So, here if you observe, the salient features are proposed by Watson and Crick. First, double helical model of DNA. The double helical structure of DNA is elucidated by Rosalind, Franklins and Williams. Franklin and Williams, they have given out the X-ray diffraction studies of double helical model. Okay. Then taking these X-ray diffraction studies into consideration, the double helical model is proposed by Watson and Crick. They have proposed the double helical model which says that single stranded DNA, 2 single stranded DNAs, here one strand will be running from 5 prime to 3 prime and the next strand will be running from 3 prime to 5 prime, which are anti-parallel to each other. So, the double stranded DNA is anti-parallel to each other. Second point is complementarity. Okay, complementarity. Complementarity, if you see, towards inner side you have nitrogen bases, right? Adenine binds with thymine via 2 hydrogen bonds. guanine binds with 3 hydrogen bonds to cytosine. Guanine binds with cytosine via 3 hydrogen bonds. Adenine binds to thymine via 2 hydrogen bonds. How the double helical model is phased? Towards outer side if you see, sugar and phosphate groups are arranged like a backbone. The backbone of the double helical DNA will be sugar phosphate groups. Internally, nitrogen bases are binded via hydrogen bonds. How they are complementary with each other? Adenine binds with thymine, guanine binds with cytosine. Like that, nitrogen bases are phased towards the inner side and sugar phosphate group is phasing towards the outer side, forming the backbone of double helical DNA. These are 2 points to be remembered. First is anti-parallel to each other, 2 strands are anti-parallel to each other. One runs from 5 prime to 3 prime, other strand runs from 3 prime to 5 prime. Second point is complementarity. Third, sugar phosphate groups are placed towards the periphery and nitrogen bonds are at the center. These are the 3 major points. Clear, right? From Watson-Crick model. Third is number in any double helical DNA, number of purines is equal to number of pyrimidines. Number of purines will be equal to number of pyrimidines. Purines will be adenine and guanine, pyrimidines will be cytosine and thymine, right? So, number of purines are equal to number of pyrimidines. Very important statement to be remembered. It can be asked in the numerical application also. Giving out any double stranded DNA, they can also give a numerical application taking out the point that number of purines are equal to number of pyrimidines. So, taking these salient features into consideration, what are they? They are 5 points. First, anti-parallel, 2 strands are anti-parallel to each other. Second, complementarity between the nitrogen bases. Third, sugar and phosphate groups are placed towards the outer end, they form the backbone of the double helical DNA. Internally, nitrogen bases are present. Fourth point, number of purines are equal to number of pyrimidines. Fifth point, a complete turn, a stable DNA is noticed as B-form DNA. Today we learn 3 forms of DNA as well. But remember, in our cells, human cells or normally functioning cells, metabolically active cells will be having B-form DNA, wherein single turn is addressed as 1 pitch. The pitch of DNA is 3.4 nanometers or 34 angstroms. 3.4 nanometers or 34 angstroms. And the distance between 2 base pairs at the center, 2 nitrogen bases adjoined via hydrogen bonds, this is addressed as a base pair. The distance between 2 base pairs here is 0.34 nanometers or 3.4 angstroms. These are the 5 major points to be remembered and addressed. First is about anti-parallel nature, second is complementarity, third will be number of purines is equal to number of pyrimidines, fourth, backbone of DNA will be sugar and phosphate group, internally the nitrogen bases are present and 2 nitrogen bases connected via hydrogen bond form a base pairs. The distance between 2 base pairs will be 3.4 angstroms or 0.34 nanometers, and a single pitch or a single turn of stable DNA will have how much length it will be? 3.4 nanometers or 34 angstroms. These are the basic points that you have to know from salient features of DNA. Analyzing the salient features of DNA, it is Chargaff who has proposed some rules which are very important. Quite a times in your NEET exam, Chargaff's rules were asked. Okay, Chargaff's rule. In 1950, Erwin Chargaff concluded that any DNA molecule, the amount of purine will be equal to the amount of pyrimidine. The amount of adenine is always equal to thymine, the amount of guanine is always equal to cytosine. So, from here he has drawn out a base ratio, that is A plus T by G plus C. It may vary from one species to other species. If I have to give you an account on this, that is adenine plus thymine by guanine plus cytosine. The ratio of this varies in between different species, adenine plus thymine by guanine plus cytosine. It can be less than 1 in prokaryotes. In prokaryotes it can be less than 1. In eukaryotes it can be more than 1, around 1.2. Fine, right? On a human DNA. This base ratio will vary. In deoxyribose, sugar and phosphate components are in equal proportions. Okay, number of sugars will be equal to number of phosphate groups. That is about Chargaff's rule. Fine. And now we will analyze 3 different forms of DNA. One is A type, second B type, third Z type. Okay. Both A and B type, the helix sense will be right-handed fashion. The helix is formed in the right-handed fashion, whereas Z DNA is left-handed, and in a single turn, just now we have seen, right, in a single turn, that is addressed as a pitch of DNA, the length will be 3.4 nanometers here, that is 34 angstroms. But how many base pairs are present in a single pitch of the DNA or one turn of the DNA? A type of DNA will have 11 base pairs, B type, the stable form of DNA, will have 10 base pairs, Z type will have 12 base pairs. Okay. These are the helices that you can see for both A, B and Z type DNAs. Okay. And major groove and minor groove. This is addressed as major groove. Yeah, can you see here, this is addressed as major groove and this is minor groove. 2 grooves are formed via double helix. When double helix is formed, you can observe 2 grooves in the double helical model. This is known as minor groove and this is major groove. Here also you can see this is minor groove and this is major groove in B form of DNA. This is minor and this is major groove in Z DNA. What type of minor and major groups you can observe in A, B and Z DNAs? A type DNA, the major group is narrow and deep. B type DNA, it is wide and deep. C type DNA, it is flat or absent. Minor groove is wide and shallow in A type DNA, this minor groove. Minor groove in B type is narrow and deep. Minor groove in Z type DNA is again narrow and deep. Next, occurrence. Where can you see A type DNA? In dehydrated DNA or normal physiological DNA? In B type DNA is normal physiological DNA. Z type DNA is observed in high salt concentrations because those are special sequences like GC, guanine, cytosine rich nitrogen bases are present. Biological relevance. B type DNA are most commonly observed in biologically active cells. Most commonly observed type of DNA in the cells are B type DNA. Clear, right? These are 3 forms of DNA. If you want, you can take a screenshot of this. Quite a times the questions are asked and all points regarding B type DNA you have to remember for sure. Okay. So far from double helical DNA, what all we have discussed? Nucleic acid is a polynucleotide chain. How a single nucleotide is formed? What are the bonds involved in a single nucleotide? How a single stranded nucleic acid is formed? How a single stranded DNA or RNA is formed? And what are the silent features of DNA? What are the Chargaff's rule? And we have also exercised 3 forms of DNA, right? So with this, the basic introduction of nucleic acid is clear. If you listen till here, you will get a complete idea on the nucleic acid, and when you are going through the chapters biomolecules and the chapter molecular basis of inheritance, it will be easy for you. Now, we will look into the concept of enzymes. Here we go with the concept of enzymes. Enzymes are very important. Every year in the NEET exam there will be at least one question on the enzymes. Enzymes are commonly noted as biocatalysts. Biocatalysts. Most of the enzymes are proteins only, but then there are few nucleic acids which also serve as enzymes, which includes, for example, ribozymes. Some nucleic acids also behave like enzymes, for example, ribozymes. Mostly all enzymes. There is a statement which is asked multiple times in the NEET and MCET stating out that all enzymes are proteins, but all proteins are not enzymes. Remember that all enzymes are protein, but all proteins are not enzymes. But then again, there are some nucleic acids which behave like enzymes, for example, ribozymes. Enzymes are known as organic catalysts. There are inorganic catalysts too, which do not occur in the living cells. Inorganic catalysts which will not occur in the living cells, and their specificity, their temperature tolerance is different. We learn the differences between organic catalysts and inorganic catalysts. Organic catalysts, also known as biocatalysts, and inorganic catalysts. So here these are the differences between the enzymes which are organic catalysts and inorganic catalysts. First, you see, enzymes are complex biomacromolecules made up of what? Proteins. Enzymes are proteins. Inorganic catalysts, they are simple small sized molecules or some elements. So this is the first structural difference. Second, these catalysts will operate only inside the living organisms, that is inside the biological systems. Inorganic catalysts are found and they function in non-living systems as well, which is outside the cell. Third, each enzyme is highly specific to a single biochemical reaction. Inorganic catalysts, a single inorganic catalyst act on multiple different chemical reactions. There is no specificity here, but here you can see every reaction is highly specific. Second, enzymes lose activity at the temperature above 40 degrees Celsius. Above 40 degrees Celsius, enzymes will lose their activity. Here inorganic catalysts, they remain stable and they work efficiently even at higher temperatures and pressure. Okay, even at high temperature and pressure, they will remain stable at work. So these are the key differences between enzymes and inorganic catalysts. Clear, right? Very important to be noted and addressed. And then there are two mechanisms of enzyme action which you have to remember, and from enzymes you have to look into the concepts of enzyme nomenclature, activation energy, transition energy, the graph, and then you have to look into the cofactors as well. These are the three concepts which we are not dealing now. Today we will deal only up to mechanisms of enzyme action. Listen carefully, ma. Enzymes from thermophilic organisms are thermally stable and they retain their catalytic activity even at higher temperatures. Even at high temperatures they will retain their catalytic activity, that is up to 60 to 90 degrees Celsius. They will make thermal stability a key feature of this enzyme. Remember that, okay? Enzymes are thermophilic, enzymes from thermophilic organisms will be thermally stable and their catalytic activity will be functional even at 80 to 90 degrees Celsius. But this is only from thermophilic organisms. This is only from the biocatalysts which are extracted from thermophilic organisms, okay? And now if you look into the mechanism of enzyme action, let me take one substrate, that is sucrose. Sucrose is the substrate which is a disaccharide. To act on sucrose, the enzyme which is there is sucrase. This is the first model which is elucidating lock and key hypothesis. Like for every lock there is a specific key, right? For every lock there is a specific key. Same the case here. The lock is assumed like an enzyme and the key will be substrate. Here substrate will fit into the enzyme and this forms a stable enzyme substrate complex. After enzyme substrate complex is formed, this turns out to be enzyme product complex once the enzymatic function is on. Enzymatic function, if it is on the substrate, this forms enzyme product complex from ES to EP. So, to transit from enzyme substrate complex to enzyme product complex, high amount of energy is required, that is known as transition energy. Normally, from a substrate to product formation without enzyme, the energy required is transition energy. But here enzyme presence is there and this is turning from ES to EP, enzyme substrate complex to enzyme product complex, and the energy required is minimal energy, which is known as activation energy. Activation energy. So, remember these two terms which are very important to learn the graph also. Graph based questions you will get, wherein here we learnt about two types of energies. One is transition energy, second is activation energy. Transition energy is the maximum energy to transit from substrate to product in the absence of enzyme. In the presence of enzyme, the energy required to transit from enzyme substrate complex to enzyme product complex, the minimal amount of energy whichever is required is known as activation energy. And at the end, the products are released like glucose and fructose, okay, from sucrose, which is a disaccharide. In the products, two monosaccharides are released, that is glucose and fructose. Clear, right? This is lock and key hypothesis, but then this is not widely accepted because this is not realistic to the biological cells. There should be some flexibility. It is not like lock and key how the biological functions, right? In biological functions, there should be some flexibility on how the model is operated, enzyme substrate to enzyme product transition. That is well explained in induced fit model. In induced fit model, you can clearly see how an enzyme substrate complex is operated. In induced fit complex, I will tell you in very simple terms. It is like hand glove model, okay? A hand which fits into glove. Any type of glove will fit to any type of hand. So, as there is more flexibility in the model, that is induced fit model, compared to lock and key hypothesis, induced fit model is widely accepted. Induced fit model is widely accepted. These are the key differences between lock and key hypothesis and induced fit model. Lock and key hypothesis is proposed by Emil Fischer. Lock and key hypothesis is proposed by Emil Fischer. Induced fit model is proposed by Daniel Koshland. This concept, enzyme's active site, is exactly complementary to substrate shape, but here active site will changes shape to fit the substrate, okay. The substrate will fit on the active site of the enzyme. Here the active site of the enzyme will be changed or modulated according to the substrate. This analogy, if you see, a key fits into the lock. This analogy, if you see, a hand fitting into the glove. Here flexibility of the enzyme. Enzyme structure is rigid and it does not change. Enzyme structure is flexible and this adjusts according to the substrate. Specificity, lock and key hypothesis is highly specific. Induced fit model specificity is moderate to high. Adaptability to the substrate, this cannot explain enzymes binding to similar shapes, structures, but this can explain binding at a range of similar substrates. As this model is more flexible and realistic to the biological cells, induced fit model is widely accepted when compared to lock and key hypothesis. Now, what are the properties of biocatalyst, that is organic catalyst? Very important. First is protein nature. I said you all enzymes are proteins, okay. And then chemical reaction. Enzymes itself, they do not initiate the reactions, but they increase the rate of the reaction. Okay, they will increase the speed, accuracy and rate of the reaction, but enzymes are not the starters of the reaction. Next is efficiency. Efficiency is equal to turnover number. What is turnover number? TON, ton, turnover number. Substrate molecules are converted to products per minute per enzyme, per a single enzyme in a single minute. How much amount of substrates are converted to products? Next, unchanged form. Enzymes remain unchanged even after the reaction. Enzyme specificity. Enzymes are highly specific to the substrates. Like for example, maltase will act on maltose, not sucrose. Sucrase acts on sucrose but not on maltose. So that substrate specific enzymes are there in the biocatalysts. Okay. So this is the whole concept that we have learnt. A complete summary of today's session. We have learnt about nucleic acids, single nucleotide, single stranded nucleotide, silent features of DNA, Chargaff's rule, and then we have looked into the enzymes, different models of enzymes, differences between organic catalyst and inorganic catalyst. We have also looked into the different forms of DNA, and at last properties of enzymes. Learning this in detail will help you easy understanding of biomolecules. From the chapter Biomolecules, we have learnt in detail of carbohydrates, basic structures and classifications of proteins, nucleic acids and enzymes. 40% of the chapter is dealt in this bridge course itself. Okay. So, utilize this all the students, and tomorrow we will start with genetics. Thank you.